to okay, we start recording to carry on the development. And now he has his own group in San Sebastian, in Iker Basque, and he's working with his group on developing bioimage analysis tool for image analysis, advanced, but also for all audiences, which is what's important for us. So Ignacio will present the spirit of BiaPy, and Danny Franco, which is a PhD student in the lab, will do a little demo. They say that you can interrupt them. So if you have any questions, so post the question in the channel, in discussion channel, or raise your hand and we'll interrupt. Also, they share their slides, so you can have them on Google Doc. And I will now leave the floor to Ignacio.
Okay, thank you very much, Bertrand. Thanks everybody for being here today. As Bertrand said, we're gonna share the floor between Danny and I. So I will make an introduction about our library through the details, how it started, how it's going, and everything. And then hopefully that will be 25, 30 minutes. And then Danny will show you how to actually use the library in a demo for typical example of instance segmentation. That would be 20 minutes or so. As Bertrand said, at any moment, please feel free to interrupt us because we will be happy to address your questions. And in any case, if you are not that type of person, at the very end, hopefully we'll have time for questions and answers. Okay, so first things first, why yet another tool and library for bioimage analysis? Well, I guess many of you suffer the same thing as I do whenever we hear about new tools, especially deep learning-based tools that came up and they look great in order to solve our specific needs and problems. But then most of the time, they don't follow these fantastic four rules or keywords that are accessibility, portability, flexibility, and reproducibility, right? So what do I mean by this? Well, when I said that our bioimage analysis workflow should be accessible, I mean that users should be able to deploy them from any computer environment with minimal time and effort, meaning that they don't have to spend hours working with people like you and technicians and trying to configure, make all the dependencies work, the drivers, the GPU, CPU, etc. They should also be portable in the sense that once they are developed, well, the users should be easily able to sift them between computer environments and then obtain exactly the same results. They should also be flexible, right? Because this is very important. We want to take those pipelines, those workflows, and then customize them for our own projects, our own data. And finally, as everything in science, it should be reproducible. No? So in these cases, we take that software and we run it on the same input data, sometimes even that the authors gave to us, we should obtain exactly the same result. And unfortunately, that's not always the case. I didn't make up these 4 keywords, these 4 rules. Actually, I read them recently in this very nice paper, Neurodesk, that came up a few days back, which is actually about workflows for analysis in neuroscience, with a very, very big initiative. So, of course, we didn't start BiaPy like this. The beginning was much more simple, and it started with Danny, that you will see later, when he was an innocent and happy master's student that came to us and proposed us to make a project to learn deep learning for biomedical images. So together with Arrate, that you may know because it's also in plenty of bioimage analysis ventures like the DeepImageJ, the Bioimage Model 2, and now the AI for Life initiative. Well, together we propose him to work on developing state-of-the-art deep neural networks for semantic segmentation on electron microscopy images. Why electron microscopy? Well, because we had some data sets, some public data sets. Actually, he worked with 4. 3 of them were public with very nice ground truth, and something handable for a master project, and also one with from some collaborators. So he implemented models and pipelines to work on these datasets using, at that time, Keras and TensorFlow. So this came up actually very nice. So Danny became our PhD student, and we published the results of this work in neuroscience, in neuroinformatics a year later. And there, Danny did what I think is a very honest work on comparing plenty of state-of-the-art solutions, but also all papers that published before on those public data sets, and try to reproduce the results and in a consistent way, making different runs of the same whole pipelines. And not surprisingly, out of the, well, plenty of papers, they didn't have even the code, so we had to re-implement them. And we asked the users, the authors for the code. Not all the time we got it, so we had to re-implement it ourselves. And plenty of times what they reported in the papers didn't coincide with our reproduced results. Some of them they did, and that was nice. And then we also came up with our own custom models and pipelines to do that segmentation that was very robust, meaning that they consistently gave the same results. So by the end of this project, we ended up with quite nice open source code because we made it public, implementing state-of-the-art deep neural networks for semantic segmentation in 2D and 3D. Okay, because some of these data sets could also be addressed in 3D, with very robust training configurations or full pipelines that consistently gave the same results. So they were reproducible. We also have data at the beginning of the pipelines to handle the images, to load them, to partition them, etc. And we started to develop specific data augmentation methods for this type of microscopy, at the time, electron microscopy. Of course, we had also in this set of code evaluation metrics to compare the methods among themselves. And also, we did something I think quite interesting that is usually not given a lot of attention. That was to compare not only the deep learning models, but also the post-processing methods that we apply to them. Because usually we create patches of the data, we pass them to the network, we obtain the results, but then the way we merge those patches and we did different strategies, then it has a big impact in the results. So we also compared that. So in other words, we ended up with a kind of Python library for semantic segmentation workflows. I say somehow a library because it was more like a bunch of nice scripts, but they were not organized in a proper way. But anyway, a year later, with some collaborators from Harvard University, we released more data, much larger data sets called MitoEM, which has one big volume from rat brain and one big one from human brain, where the purpose was to do instance segmentation as opposed to semantic segmentation. Here, we also needed to recognize each individual mitochondria object in 3D. So that means that we had to create new models, new data augmentation, use different post-processing strategies, because now we had to obtain the instances out of the probability maps, for example, of the mitochondria. We have to evaluate the model in a different way with new metrics, and of course adapt the code that we had for larger scale, because those datasets were an order of magnitude larger than what we had in our previous work. So suddenly, our nice set of scripts became somehow a chaotic set of not only Python, but other languages scripts. So at this point, we sat down with Danny and said, okay, we have here some identified needs that we have to address. First of all, we wanted to build a proper library that was easy to install, because we were changing sometimes computing environments and it was tough for us to develop, and maintain, but also for other people to contribute to, if we want this to be reused. And that was one of the main focus because reusability was very important for us. We started to have other projects, and we didn't want to, for every project, copy paste the previous code and adapt them on the fly, right? And also we were starting to have other problems in terms of bioimage analysis tasks. We wanted to incorporate, for example, image classification workflows, super resolution workflows, object detection in 2D and 3D, especially for detecting the center points of the object of interest, or having, for example, image reconstruction or denoising type of task. And the most important thing for a library, we needed to find a proper name, because so far it was called, the repository was called electron segmentation or something like that. So this is how we came up with BiaPy, that stands for Bioimage Analysis Pipelines in Python, that we already introduced to the community last year at ISB. So the main building blocks of BiaPy are these. So we wanted to create something for both users and developers, taking into account our own community, the bioimage analysis community. So meaning, as you may know, people that work usually with microscopes, and they need to process their data, and hopefully they know how to handle a little bit the computers, but they are not so familiar with installing plenty of things, right? And then we were also targeting people like us that work in a computer science environment, that we develop methods, and we could adapt them to the specific bioimage analysis workflows. And of course, in bioimage analysis, in microscopy, we had to have solutions for this type of data, which is usually 2D, 3D, multi-channel, that is many times very large, in either in a single dimension or in many dimensions. Or we have also set of volumes that we had to process. So we had to create a library that was adapted for all those possible inputs. As I said before, we were starting to face different bioimage analysis tasks, not only segmentation, which is, of course, very important, but we started to collaborate with other people and target many other tasks. So for this, we created a library with a backend in Python. Very recently, we have migrated from TensorFlow to Pytorch, just because we have seen that many available solutions in terms of deep learning in academia are more available in Pytorch, so it was easier for us to reuse them. We have become community partners with the bioimage model zoo, which means that we can, from the zoo, we can consume their models and full workflows, okay? And we can also export our workflows into the zoo. Because we are because we are developing in Pytorch, we also have access to the Torch Vision models, which are state-of-the-art models for different tasks in general in computer vision that we can adapt for our specific task. And of course, we can edit and customize our own workflows. And finally, as I said, we were targeting many different type of possible users of our library, our platform. So we created a front end that has actually a bunch of options. So from the most non-expert novice expertise person to the most, let's say, hardcore developer person, we have first developed a graphical user interface, which is a way of easily installing the library and all its dependencies. Also, it takes care of the recognizing the GPU, as Danny will show you. You have one, you don't have a GPU. Don't worry, it will work on CPU, it will be slower, but you can work with it. And then you can create your workflows directly through the GUI. You will see this with Danny in a moment. If you are more familiar with, for example, the zero-cost DL4MIC notebooks, these zero-code notebooks, we have also developed a version of every workflow in Google Colab. So you have zero-code notebooks where you can run experiments. Of course, it's nice because you have the GPU that Google provides, but at the same time, you are limited in terms of data size by the platform. But you can also use it to prototype, and then, as you will see, create workflows that you can later use somewhere else. For example, you can export those notebooks to a local environment and start working with the notebooks in your own computer. You can install everything through Docker. So we have a container that has all of these dependencies. Actually, the graphical user interface is doing that under the hood to install everything. Or if you are a hardcore developer, you can, of course, go to the good old terminal, go to our repository, clone it, and start working with it. So we're talking a lot about the workflow, but how do we define a workflow in BiaPy? We wanted to keep it simple. So we had three main components. Okay, at the core, it's always a deep learning model. This library is all based on deep learning approaches. And then we have a pre-processing step and a post-processing step. So the input gets into the pre-processing step, gets used for the deep learning model, either by training it or applying it. Okay, and then it gets post-processed, and then we get the outputs. What kind of inputs? Well, of course, we have images, right? So I think it's mandatory. We always have an image as an input, but then we may also have the corresponding labels. For example, if we are doing segmentation, the semantic or the instance segmentation labels in the form of another image, or even a CSV file, for example, for detection, where we have the coordinates of all the objects or the classes or the categories that we want to classify this set of images. When I put an icon of an image, but of course the input is a whole folder with all our images, or in 2D, 3D, etc. And for output, it may be that we obtain an image. For example, if we're doing segmentation, we get the labels, or we're doing denoising, we get the denoise image, for example. Or we may get the CSV file with the corresponding positions of all the points that are predicted in a detection workflow, or for example, the categories of the classes. In pre-processing, we're putting all the data handling, but also how we prepare the data to feed it to the model, no? In terms of doing patches, normalizations of the intensities, or even adapt, as we will see later, the problem representation, so it's easier for the model to learn how to solve it. And for post-processing, well, sometimes the raw outputs of the models are not enough, so we have to do some filtering, or we have to, as I said at the beginning, create the mosaic of all the patches together, or, for example, suppress all the detections that are nearby. Yes, Nicola?
What are the format of the image you have to put for the pre-processing, and what are the format of the image of the output of the processing?
That's, I think, it's next slide.
Okay.
Good. I'll show you. So, well, this structure for us is important because we think that it has some advantages for users. It's easy to then play with the different pre-processing, post-processing methods, or even interchange models and see which one works better for them, right? And for developers, it also gives them freedom, so they can contribute to a specific part of the workflow and not a full workflow at a time. No? They may be very good at creating new deep learning models, and then they can contribute a model, or they can contribute a new image processing step for pre or post-processing. So this could be everything that is going on under the hood, okay? I will answer Nicola's question here. So from a user perspective, no, depending on the expertise of that person, then in the end, we're going to create a workflow, and everything is going to be stored in a single file, in a configuration file that has this extension, YAML file. I will see next. And here we describe the inputs. The inputs are for images, very typical formats. They could be TIFF, PNG, etc. But we have also the NumPy, H5, and very recently we have included SAR, so we can use very large data sets and load them in an efficient way into memory without collapsing our RAM. So this is very important for more or less large scale processing. So these images are input into the workflow. And as we said, there are 3 steps: preprocessing, deep learning, training, or inference, the application of the model, and then post-processing. Everything is done in a memory efficient way, which is transparent to the user. And if you have, for example, multi-GPUs, then you can use them. As I said, if you have a GPU, everything's going to go faster. But if not, it's okay. Training is very slow, usually without GPUs, but inference, the application of the model, is quite fast. And then as output, then you get images that you can store. I mean, everything goes stored, as Danny will show you, in a folder. You have these CSV files, maybe label images, etc. And very important, then you also get the model that you train. If you are doing training, you get especially a file with the weights of that model. This is important because when you want to share this workflow, then you will have to share the configuration file, but also the weights of the model. These 2 files, okay? If you just want to start from one configuration and do the training yourself, you don't need the train model. But if you want some collaborators to use your own workflow with your specific model, then you have to share both. Okay? And then, of course, depending on the task, you may also get a bunch of metrics and measurements of your result. I mentioned it a little bit at the beginning, but one special thing of the library is that we have developed microscopy-specific data augmentation, meaning that we have adapted methods that are very popular for natural images to increase the robustness of our models. For example, by masking areas of the images, blurring them, adding noise. This works in 2D and 3D. So the models have to not only solve their own tasks, but also deal with this problem, which makes them usually generalize better. If you work with images or volumes that are sections, then we are also providing ways of simulating misalignments or missing sections. So even if they are not in the original dataset, the training dataset, the model can be ready for that. And also we have some tools for color images. Okay, so how do we configure a workflow? As I said, everything is in a single file. It's called a YAML file, which is a text file. It has no code, but it has some keywords, you see in blue, where they define exactly the parts of the workflow that I defined before. For example, the type of task that you're doing, where do you have your data, how you're going to do the splitting of the data, where you're going to store the results in the post-processing step, etc. And then, of course, a lot of parameters about the deep learning model. So we provide for every task, let's say segmentation, detection, super resolution, etc., one template, so you don't have to start from zero, and for both 2D and 3D. So this way, there's plenty of advantages. You can very easily prototype. You don't need to code. You just go and change. If you are a hard code person, well, the keywords here. But you can say, yeah, but I don't know the keywords. Or I mean, even if I start from a template, okay, I can change the folders, but some of them, they may escape me, right? No, don't worry, because that's not the purpose of BiaPy. So many times we don't do this because we have other options. For example, let's say instead of semantic segmentation, you have instant segmentation task, as Danny will show you later, no, in 2D. And then there are some keywords like this that may not mean anything for you as a human reader, right? So you can either go to the notebooks. In the notebooks, everything is explained in human language, right? And then under the hood, it will create this file. For example, this one, as you will learn later, corresponds to the problem representation that can be extracting from the objects, the binary mass, the contours, the distance map, etc. Or even better, as Danny will show you, you can use the GUI. This is the easiest thing, because in the GUI, I don't want to get into details because Danny will show you next, but you have, well, apart from recognizing directly if everything is installed correctly or you have to do it, you have three main options, which is create a new workflow from scratch, okay? And then you can select what the task, and then it will load you the template. Okay. You can modify an existing workflow. You load it from file, and then you modify it. Or if someone gives you the existing workflow and hopefully the weights of the model, you can just click on run the workflow, and you will load both and apply it to your data. So what kind of task are we targeting here? Of course, we started with a lot of segmentation. So in decreasing order of development, we have a lot of options for semantic segmentation and instant segmentation. We have worked a lot as well on detection, especially anisotropic 3D data object detection. Okay? We have lately worked quite a bit on single image super resolution, meaning that we, from low resolution images in 2D or 3D, we can produce images that are increased in resolution by a factor of 2 or 4 or 8. Well, something very specific of BiaPy, which is pre-training our models using self-supervised learning, meaning that we showed our models images without labels, and then we can pre-train them, for example, with a specific task that don't require labels, for example, denoising or even super resolution. We can simulate that. And then once we have that trained model, we can plug it into any of the other tasks, and it will work usually better because it has already seen that type of images. And then we have, as I said, denoising and image classification workflows. So summarizing, for every pipeline or every workflow, we provide 2D and 3D templates. We have a full documentation site that you can click here and go to it. We provide notebooks that are runnable in Google Colab, and they have real data. For every notebook, we provide data's public data set that you can run and reproduce what is supposed to happen. And in some cases, we also provide full tutorials, very detailed tutorials that reproduce what we have already published. Okay, so in some of course, we use BiaPy almost for everything now. And then in our publications, we provide these workflows. So in some cases, we have even more documentation about that. So as conclusions, I hope I convince you that we have now created ready to use all-in-one library for bioimage analysis that uses deep learning that is portable and reproducible, thanks to use this system of containers. It's also flexible, so you can customize the workflows easily because you can take the templates and adapt them, or you can create them from scratch. The easiest thing is to use the GUI, as Danny will show next. And it's oriented to all audiences, from very inexperienced users to hardcore developers. I hope this is something that you are convinced of. We are providing all these front-end options that with this target at least. So it's making it accessible. They of course, is not only including things that are already there, but we are creating our own specific solutions for like microscopy, electron microscopy images from basically all our collaborations. And we also have, remember, these data augmentation techniques for that. And very important, you have to remember that this is under continuous development. So be nice to us if you use it and you have problems, please just any feedback would be super welcome. In our to-do list, we are now creating a landing page because so far we have the documentation site, the repository for the GUI, the repository for the main code, the documentation, etc. So we want to improve a lot of the documentation. We also want to create more tutorials and notebooks, not only for the workflows, but also to show you the different options for data augmentation or pre-processing with real examples. And one thing that we really want to do is creating more videos of all of this. We would like as well for the developer point of view to create a plugin system, so it's easier for them to contribute to this. It's so far you have to know about the API of BiaPy to do it, and we think also that it makes total sense to use Napari for visualization, because so far the inputs are folders with images, the outputs is another folder with all the results that you get, images, files, etc. But for visualization, we usually use Fiji, as you will see next with Dani. And finally, we keep trying to increase, and I think we're getting there, our user and our developer base. So now, to introduce you Dani's part, I'm gonna explain how we do instance segmentation in BiaPy. Okay, in BiaPy, what we have is what we call a generic bottom-up approach to instance segmentation, meaning that instead of out of the input image obtain directly the instance segmentation results with the model, we divide this in steps. So in our image is input to a model in 2D or 3D, depending on the data, and we have what we call the problem representation here, meaning that okay, we may make the model not only output, for example, the object probabilities, but also their borders, or even a kind of distance map representation. So these three outputs, these three channels, are very useful in order to identify later with some post-processing every instance. And how do we do this? No? Well, we can use our good old friend Waterset. We have other options there. So we can combine the outputs into a single instance image. Let's see this example. These are slices from a 3D result. No, we're trying to identify these cells here. So one output channel will be the probability maps of every cell, which, as you see, is not perfect. Okay. We also have another channel that is showing us the probability maps of the borders. Okay. And then we are also in this problem representation calculating a distance map from the center pixels or voxels of the objects to the borders. Okay? As you see, it's not perfect. No, we have a lot of garbage inside this area here. This is transparent to the user because what we provide BiaPy is just the raw images and the label images with instances for training. So what do we do here is that, okay, using some simple thresholding that is implemented there, we can calculate the mask, general mask of where the objects are, okay, based on these probabilities, and then combine the other channels to get markers that are separated thanks to the contour images from each other. And then this combined with the inverse distance of the, for example, this is just one option of the distance map, we can run marker control Waterset and obtain the final segmentation in 2D or 3D. Okay, so now it's demo time. So I'm gonna give the floor to Dani, and he will show you.
Yeah. Thank you very much, Ignacio. I don't know if there's questions for Ignacio before Dani show us the tool that looks very exciting. And
yeah, maybe one
Romain,
question.
Your mic is a bit low.
Sorry.
Okay, that's better.
Thank you, Ignacio, for the nice talk. do you see it mostly for workstation, people installing Docker desktop, downloading the GUI and starting? Or do you also see a use case with GPU grids, like a 200 GPU somewhere, and you can use the Docker image there and forward in the web browser or something similar?
So far we have used it for workstations, but also and actually Danny works in a data center, and we have nodes with many GPUs, and we are using them there. So
Okay.
we have it's not adapted. I would say, Danny, correct me if I'm wrong. It's not adapted to use directly, for example, with a queue system, but I think in a data center you can install it in nodes and then use it there.
Okay, and you have documentation about this part too, or it's something undergoing and it will be soon documented?
Well, the documentation is there. We have, well, if you want to run this through a cluster system, through a queue system, you need to use command line. So yeah, those options, if you run it through the command line, or if you use the graphical user interface through a workstation, both things are documented. Yeah.
Cool, thanks.
Otherwise, you can always pick Danny and
yeah.
yeah,
There is
Nicolas?
another question. Yeah.
Yeah.
Yes, a different question about the workflow itself. So you showed that you integrated deep learning into for the segmentation, but could we implement some machine learning pixel classification models into, for example?
So you mean if yeah, for doing segmentation, I guess.
Exactly, and or for example, even object classification after.
Yes. Yeah. So far we don't have anything like this. So for segmentation, we use segmentation models. And for classification of detected objects, we also have our own models. But yeah, I guess it should be implemented as a post-processing step, no? So create something, a workflow that is larger than these 3 steps.
It just because I don't know how you manage hardware resource, but I think if we want to do some 3D deep learning training model, we need a very good graphic card to do such training. And with machine learning, we can have less needs of power for doing some good things. And I'm sure you have very good tool for segmentation after to help classification, doing some good object creation and then object.
Well, we actually use kind of regular GPUs, and in our experience with 3D, for example, segmentation, we didn't have to use very large networks. So that's allow us to use the regular pipelines, and then using this patching system is not a problem for the size of the data. So in that sense, I believe that even if the data is large in 3D, we can still live with that.
Cool. And I see some cool model that use just a few annotation and not all the image annotated to do some pretty good segmentation. I think here we need a full image annotation yet, or do you have some deep learning model that can have just few annotation for that?
Yeah. So far we do full annotations.
Yeah.
Yeah. This is something that it's a matter of adapting the workflow. If the deep learning model can be set there, I don't know which model you are referring to, but let's say it is implemented in Pytorch, then it's very easy to just plug it into our workflow.
Okay.
And then the input data, we don't care how it is. It's the one that is
Okay.
It matches the model. What we don't have is an interface, for example, to select data for the training of that model. That should be done somewhere else. So far, as I said, all the data is input through a folder.
Yes, but we have the output. We see the output, and we see, okay, the output is weak at this part. And what I see for the presentation of this kind of model is say, okay, it's because I don't put some context of the few annotation I've made, so they just annotate a little more around other annotation, and then it improve. But they have the same way as you. They do the training, they see the output is not very satisfying, and they redo the training. So this is something that could be
I guess
interesting.
that type of let's say interactive learning approach that requires usually to have an interface, so you can go back and forth showing results, retraining, and improving the annotations. So this is more thought to have batch processing. So if you have already that model, you can input it there. I mean, you could also work in a way that you do the processing in a workflow like this. You check the results, but then you have to re-annotate somewhere else.
Yeah. Okay, thank you.
Okay,
thank you.
I don't think there is more questions, so I guess Danny now is going to move on to the demo.
Yeah, so I will share my screen. Can you see my screen? Yeah. Yeah, okay. So as Nacho has said, I'm going to do a demo for instant segmentation workflow, which means that we want to, well, I open these two images here in Fiji. So this image of the left is the raw image where some nuclei are there. And here on the right, we have the label image. So we can, if you want, apply a LUT so we can see better. But this is the problem. Actually, the instant segmentation problem where we want to segment each nuclei with a different or unique integer, as you see. Okay, so the first thing we are going to do this with BiaPy and using the graphical user interface. So the first thing that we need is to go to our page of the project of BiaPy. As Nacho has said, we are still doing a landing page. But for now, you need to go to this GitHub page. Okay, so here we have in the README, we have here a section where you can download the binaries to install the graphical user interface. So you can just click, in my case, I'm using Linux, so I can download the Linux binary. It's going to redirect you to the drive to download it. So I'm not going to do that because I have here the binary already, but so you are aware of how you can download the BiaPy graphical user interface. So here I have the binary. You can just double click it. Well, actually here in Linux, we are still working to put an icon here because it's not a trivial thing, I think. So, but in Windows and in Mac, you will see the icon of BiaPy, okay? So just when you double click it, you are going to see this graphical user interface, as Nacho has introduced a bit. So you can move the graphical user interface by clicking on the logo or the empty spaces over here. Okay, so here on the top we have a few external links, the project page with which redirects you to our project page that we have just seen. We have a forum. Also, we have joined to the forum and see to discuss anything related to BiaPy. We have also the documentation, templates if you want to use them, the collab notebooks that we have implemented, and then there is a citation also link that you can use. Okay, and there, as you can see, we have in this main window we have two main dependencies check. One is to check whether you have Docker installed or not. So this is gonna be green when you have Docker installed. Let's say in Windows you need to install the Docker desktop, and in Linux you need to just install Docker through the terminal or whatever other kind of flag that you want to follow. So and apart from that, you have also another dependency check, which is the GPU, as Nacho has said. This will speed up the process, but it's not mandatory. For training is, I mean, the training is gonna be very slow, but the inference or applying the model that is already trained is not going to take that much time. So apart from that, we have also here a few tabs on the left where all the tabs are related to create the configuration file that is going to be the input for BiaPy, for running BiaPy. Okay, so we have here 3 buttons. The last button is going to redirect us to the last window, which is run workflow window, and then the 2 buttons above are for creating a new workflow from scratch or loading already existing workflow that you want to reuse, or if some collaborator sends you a workflow, you can reuse that workflow by clicking in this button. Okay, so we can just go through the create new workflow button. So if we click it, we are going to be redirected to the first window of creating the workflow, which is select the different workflow that you are going to use. So here by default is the first one is instant segmentation, but you can choose another different workflows that we have already implemented. So like semantic segmentation, classification, self-supervised learning, super resolution, image denoising, detection, or instant segmentation again. Okay, so in this case, in this window, if you are not very familiarized with all the workflows that you can apply, you have here also a learn more button where you can click, and here is gonna show a full description or a brief description of the workflow. So you are more so you can read it. And here also there is another button to our documentation of this specific workflow, and that is going to redirect you to the documentation. And here you have also a few ready to use examples related to the workflow that you have just selected. By the way, we are going to use this example here, which is this one of the nuclei that I just described. So you can, in case you wanna follow the same steps that I'm going to apply here, you can download the dataset here or the template to use it. Okay, so just so you are aware of this button also. So we can just select instant segmentation workflow, and then you can continue, or you can click in the tab to go to the next window. Okay, so here the next window is a window to configure a few generic options. So the first thing to say here is that in all the windows over here we have a few basic configuration variables that we need to set, and then we have also another more advanced option for more expertise users. So we can just leave every single variable with the default settings, because they are going to because the default settings are is gonna run good with good results. So you don't need to just modify every single variable that we have. Okay, so here in the first generic window, you have the image dimensionality. Well, here you have 2D or 3D. In this case, we are going to use 2D images, so we can just leave it as 2D. Then you need to put a path where the configuration file is gonna be saved. So I can just click in a folder here. Then we need to put a name to the configuration file. Let's say segmentation experiment, whatever you want. So the last option here is just if you have some model already trained, so you can apply it or fine tune. Okay, so we can, as we are training from scratch, we don't have any, so we can leave it as no. And in all the variables also, we have a button of information where if you put the cursor, a message of a more detailed description of the variable, it's gonna pop up, so you can see what is doing all the variables, what are doing. So we can just click on continue because we have just filled this, and then the next two windows are gonna be the windows for the training data and for configuring the training data and the test data. So here again we have a few variables that we need to set, and there is also another advanced section that you can go through. Okay, but here with lots of data augmentation methods are here. Also, if you put to yes, there are lots of them. So we can, just for the sake of the example that we are doing here, we can just leave it as it is. Okay, because with the default values, it's gonna perform good. So here you need to set the raw image folder, the training data folder, so we can just browse it. I have already here prepared the folders, so I can go to them directly. And you need to just select the folder where all the raw images, let's say these images, are stored. Okay, so we can just have this path, and then well, in my case I will copy paste the same path, because on the same path in a folder that is called the labels are going to be there, but you can browse it. Okay, again, and here are the annotations, which are these images, this image over here. Okay, so we have just configured the training data paths. So we need to set a validation data. So the model training is not overfitting or learning too much the training data. So we can extract a part of the training data to use it as validation. And we have also here a few more parameters that we can configure. Let's say here we have the model that we are going to use to solve the task, which is unit, in this case, a regular unit. Okay, so apart from this tab of general options, we have also workflow specific options or variables that you can select. I wanna leave them as they are, but you have these options are going to change depending on the workflow that you have selected. Okay, so we can just leave it as it is, and then continue. We want to go to the next step, to the next window, which is configure the test phase, and this here there are a few variables, options, basic options, and then a few advanced options again. So the first thing that we need to set is the path to the images. So we are going to here select another path which with images that are not used for the training. So I will choose them. And then also you have the option here to select whether you have the annotations or not. In this case, I want to leave it as no, but you can select to yes if you have. And in that case, some metrics are going to be calculated so you can evaluate the model performance on the output of the model. Okay, so and in this window we have the last button that is not going to have a continue text, but create a configuration file, because it's the last step to create the configuration file. So if you click it, you have already selected all the things needed. So the configuration file is gonna be created in the path that you have just selected. Okay, so as we have created through the windows the configuration file, it will redirect you to the window to run BiaPy, and as we have just created a new one, it will be loaded by default as you are seeing here. So we just need to select an output folder. I can just put an output folder. We have here also an option to check the file, whether if there is any error with the file or not. We have everything correct. And so we can finally run BiaPy, run the workflow. Okay, so another window is going to appear. Here there is a previous step of downloading the BiaPy Docker container, but as I have already downloaded in my computer, it's the training phase is going to start, or BiaPy is going to be run directly. Okay, and here you have a few information to check. You have the date where the date of the experiment, the job name that you have selected. You have the configuration file that you have used for running this experiment. You have a few logs and the output folder, and also you have here a few bars where you can see not by looking the raw output, but if you have selected the training and the test phase, so you are going to see here how many epochs we selected to train 100 epochs. So and now it's training 1 epoch out of 100. Okay, so you can see it here more easily. So I'm not going to wait until this is complete, because I have already the outputs prepared. So I'm gonna stop the container. You can see here the status of the container state. So I'm gonna exit this, and then the output is gonna be something like I'm going to show now. Okay, so this is a the raw predictions of the image of the model, and here I'm going to show you I'm going to show you the prediction of the model. Okay, so you will have of those test images that you have just selected to be the test, you will have something like the final labels. Okay. And another thing that I'm going to show you is to modify and load a jam configuration file when you have already done the training before. So for instance, if you have a collaborator and you want to share the configuration file with the collaborator.
Danny, can I interrupt?
Yeah, sure.
A quick question about the log files. Do you log the list of images that have been used for the training? Because that's useful to be able to track that.
You have the full configuration file print in the first lines of the log, and there you have the folder that you are using for training.
Okay, the folder, but not the content, not the list of a log with all the image name and file name that you've been using for training.
No, not that. No.
Okay, thank you.
And I have a question too about do you have some loss curve and metrics output, or because you show us the result, but do we have the loss curve of training and validation and some metrics output like intersection over union or anything like that?
Yes. So as I said here in the test phase, when you are configuring, you have the option to select whether if you have the labels or not. So in this case, as I select to no, that's why we don't have any evaluation metric. But if you select to yes, then the output can be compared with those annotations that you have already. And then you can some metrics are going to be print depending on the workflow that you have selected.
Yeah, Danny, sorry to interrupt, but also I think Nicolas refers to the training, and yes, the training curves are saved, right, in the folders.
Yeah.
So all those curves and those plots are also part of the output.
Yeah. Okay, another question. Do you have any other question? I'm not seeing you guys, so I don't know if there is any. Oh, it's all good. Okay. So the next thing that I'm going to show you is when you have another collaborator, you have, for instance, train a model, and you want to share that model knowledge with a collaborator, so they can apply their own images to segment or to do whatever workflow you have selected. So the things that you need to send them are two files. One is the configuration file that you have just used, and another one is, as Nacho commented out, are the weights of the model. Okay, so here we have two files. So in this case, you can load and modify a workflow. Let's take the configuration file that you have sent to your collaborators, then a message of the configuration file that the configuration file has been loaded into the graphical user interface is going to appear as you see. Then okay, and then you will have the same steps of creating a new configuration file, but from the loading all the variables that they are already in the configuration file. Okay, so here, for instance, you need to modify a few things. The most common ones that you will need to configure are the paths, because the paths of your collaborator maybe are different than yours, so you need to check that. And also the model weights you need to tell BiaPy where are stored the configuration, the weights of the model. So here, if I go to generic options, you have here the configuration directory and the configuration file that you have loaded. And here you have the options to load those weights that you have already trained. So if you select that to yes, then you can browse the model weights by selecting the weights of the model file. So, and then you can go to continue. Then as the training has been already done, this will be set to no, because the train you don't need to retrain again, no. But if you wanna do it, you can just select to yes and then do it again. But well, if you are sharing your model, normally it will be set to no. Okay, and then in the test window, you need to put here your data, your new data that you want to apply. So here, as I'm reusing the same configuration file, this path is gonna exist in my computer. But if not, you just need to browse the directory where all your images are there, and you can leave the rest as it is. Okay, so then you can create the configuration file. Then if you didn't change the name of the experiment or of the file, it's gonna pop up a message that you are overwriting the configuration file. So we can just say to yes, or if you wanna keep your version of the configuration file, you can go to change the configuration file name here. Let's say test or whatever. Then if you go through the windows again, then you can create, and then it's gonna create with a different name without overlapping the configuration file. Okay, so we can just click in OK. You can check again the file. Some errors appear that the test path is not existing. Well, I will check it. If we go to the test, I can put the path. Okay, now yes. And then we can create again the configuration file. We can overwrite it. Then put yes. Configuration file has been just created, so we can select to OK. And then everything is loaded. We can check the file again, so no errors are here. I thought I was using the same configuration file, but I think that I used another one, so that's why that path was missing. But then we did it again, and then now that the path is correct, here the check is in green, so we can run BiaPy, and it's gonna run with the new information that we have just selected. And now it's doing the inference only. Okay, so I will just stop this to not bother you with the output. And these are the two things that we wanted you to show. So if you have any question, or Nacho, if you want to add something else.
Sorry, I was a bit late, so maybe you already answered, but do you have a workflow that can do segmentation and classification at the same time?
No, okay. Not yet, no. But you can do both things. If you can segment the cells or whatever object you wanna segment, you can then prop maybe a region of that segmented object, and then pass through the classification workflow.
Yeah, but if I do the classification workflow, it will be done on the mask. No?
No, you can crop a region of the
Okay.
raw image, not
Okay.
the label. Yeah, but with the
Okay.
label, you have already segmented the cell, and then you know
Okay.
the position to make the crop, and then use
Okay.
the classification. Yeah.
Yeah, this is actually in our to-do list, because since we are incorporating all the models from Torch Vision, some of those models, they do exactly as you say.
Thank you very much.
I have another question about the Docker itself. Just with my little experience, I tried to install a deep learning formic everywhere with the Docker. It was not very easy, but I finally make it. And my problem is that when I launch the use of the Docker itself with a notebook, it downloads everything. It takes gigabit of data, but it was on my OS system, which has only 500 gigabit of for the OS, and it filled all my hard drive. And I was saying, okay, if I want to do such Docker things, maybe I need to buy a new hard drive with 2 terabytes of data. How does it work here? Could we say that, okay, the Docker is here, but please do put the model and everything on a very huge hard drive, which is not the OS hard drive, or is it the same problem that I need to buy a very big hard drive? Or it's me that I don't do the good settings, maybe.
Yeah. So yes, well, in our case, the Docker container, it's around 15 gigabytes. And when you reach this window, you will see here like a message with how the size of the container. But yeah, sure. I don't know. We can have just when you run the workflow, we can have just like an information message. So, okay, you are going to run this, and you are going to download automatically the Docker container. So be aware that you are going to consume like 15 gigabytes or whatever. So the user is aware of that also, yeah.
Just to mention, Nicolas, that in the Docker desktop application, you can define the folder where you want your Docker images to be saved, and you can define which hard drive you will be using. So it's not on the C.
Cool. Okay. So I'm not I end user, so I think I miss these things. Okay, but having the idea of on my size, I need to download it. It's still interesting. Thank you, Danny and Roma.
And I think once you will have downloaded once, you don't need to download it anymore. It's there unless you make some room on your laptop and then you delete the file. But otherwise, and everything which is common between different Docker will be shared, if I understood correctly, the Docker mechanism. So if a new version bugs fix some stuff, you don't need the extra, I don't know, 15 giga, maybe just 1 or 2 more would be enough.
Yeah, that's true. Yeah, because we have named all the containers like the latest container. We are going always uploading the same container with the same name. So the graphical user interface is gonna check if there is some new container than your local container, and it's gonna download it automatically and overlap the last container that you have. Yeah.
Just
And
maybe for
also,
versioning, sorry, just for versioning sake, it might be interesting to have a document somewhere where you have the release date, so people can know, okay, at this time latest was version 1, version 2, version 3, and so on. So even if you always use the latest, if we want to enforce a certain one, we can still do it.
Yeah. You're right. Yes. At this moment, we have in Docker Hub all the containers that we have upload. But it's true that maybe we need to put in a documentation clearly the versions that we have upload, the version of the container that you are using in this graphical user interface. So you are aware of that also, yeah. And also an interesting thing here is that we are supporting a few versions of NVIDIA drivers, CUDA, let's say CUDA versions. So maybe all we have here, we are using here 11.8 CUDA version, but we have also another container with 10.2 version of CUDA. So NVIDIA cards, old NVIDIA cards, let's say, can also use BiaPy.
Danny, I have another question regarding your Docker images. Just had a look to the documentation, and in the command line part, you mentioned installation is doable with a pip install and so on.
Yeah.
So I guess in your Docker image, you have an opt conda/env with BiaPy. That's correct? That you are starting. It's a conda environment that you are starting within this Docker.
Yes.
Okay. So the question behind is just I would like to do a multi-stage to add BiaPy to my own Docker, so I can just copy paste it from your image to mine, and it will work too.
That's
awesome.
Yeah. Right now we are not using the pip install approach to install BiaPy into the environment that you create in the Docker file, but cloning the environment, cloning the repository, and then doing a pip install with all the dependencies that you need. But we need to change that, yes. But you can also reuse the steps that we follow to build the Docker container in yours. Yeah, of course.
Cool, thanks.
Okay. Is there more question? Okay, it doesn't seem to be more questions. Thank you very much, Ignacio and Danny. Very exciting. I guess you may see a burst of download on your GitHub page, hopefully, and then try out. It looks to me kind of solving the issue I had with Google Colab that's break down every every 6 months. And having something that's out of Google Colab with Docker or your own GPU, it's great. Okay, so thank you very much for your effort, and I guess we'll see you again on the RTmfm little webinar, and probably be in touch via your GitHub page or trying your new tool. Thank you very much.
Thank you so much, everybody, for being here today and staying until that late. Enjoy your lunch.
Thank you. Bye-bye.
Thank you very much.
We'll send you the recording. The recording will be available via either Ignacio's team or via the RTmfm. See you guys. Have a nice weekend.
You too. Take care.
You too. Bye-bye.
Thank you. Bye.